Viral replication is a more complex process. So let's make it simple. First, remember the steps: attachment, penetration, uncoating, biosynthesis, assembly, maturation, and finally release. First, we'll start with attachment. The first and the most specific step involves a lot of receptor interactions. These receptors are placed on the envelopes or the capsids of the virus, and they will bind to the complementary receptor sites on the host cell surface. Examples: you have the HIV virus, which has a surface GP120 receptor binding to CD4 molecules on the host cells. Next comes the process of penetration. This process can happen in three different ways. One is a phagocytosis, or in viruses what we call as viropexis. It's a receptor mediated endocytosis, means the virus particle is as such taken inside the host cytoplasm. The second method is membrane fusion, where some of the viruses like HIV, which are enveloped viruses, they will fuse with the plasma membrane of the host cell, so that the nucleocapsid alone will enter the cytoplasm and the envelope of the virus will remain attached to the host cell membrane. The third type is an injection of nucleic acid, which happens in cases of bacteriophages, where the whole bacteriophage cannot enter inside. So it will just inject the nucleic acid alone inside the host cell. Next, we have the process of uncoating, which happens by the action of lysosomal enzymes of the host cell. The viral capsid gets separated from the nucleic acid, which is slowly released into the cytoplasm of the host cell. This is followed by biosynthesis, where the components required for the future viruses like nucleic acid, capsid protein, enzymes required for various stages of viral replication, and regulatory proteins to shut down the host cell metabolism are produced. This process differs in various viruses. In case of a DNA virus, the paternal DNA is now first transcribes the early mRNAs for formation of early proteins. Then there is a formation of late proteins, and they are all assembled together in the form of daughter virions. In cases of RNA viruses, if it is a positive sense RNA virus, the genome itself acts as an mRNA, which is translated to form the ribosomes. In the case of a negative sense RNA, the genome acts as a template which can directly undergo replication. The retroviruses here need a special mention, where the single-stranded RNA is converted into a DNA RNA hybrid in the effect of RNA dependent DNA polymerase, which is because of the reverse transcriptase enzyme. Now that proceeds to form a single stranded DNA, and again in the action of the DNA polymerase activity, it forms double stranded DNA, which will now transported to the host nucleus. With the effect of viral integrase, it will get joined with the host chromosome and it will stay there. So whenever there is a need, it can be translated to mRNA and to viral proteins and further daughter virions. In the next stage, all this viral nucleic acid and proteins that are formed are packed together to form the progeny viruses, that is your nucleus and capsid together. It can happen either in the host cell nucleus or cytoplasm depending on the virus. The number of virus particles which are produced are packed properly and they mature, and finally comes the release. There can be two types. One is with a lysis of the host cell, that most commonly happens in non-enveloped viruses and bacteriophages, or it can be through budding through the host cell membrane, which happens mostly because due to enveloped viruses, where during budding these viruses will take a part of the host cell membrane to form the lipid part of their envelopes and come out of the host cell to attack newer cells. So this is the general scheme of viral replication. So always remember, this starts with attachment, penetration, uncoating, biosynthesis, assembly, maturation, and finally release. I hope this video was short and easy to understand. Please do like and share this video. Subscribe to my channel for more interesting content. And if you are interested only in shorts, you can go to my shorts channel, Micro Quick, where I'll be posting only shorts. Thank you.